Hello students. Today we will start second chapter of unit reproduction, that is sexual reproduction in flowering plants. A flower. A flower is a symbol of human feelings like love, happiness, affection, mourning, sorrow, grief. But to a biologist, flower is a morphological and embryological structure and is the site of sexual reproduction. So let's have a quick review of parts of flower. As we have already learnt in earlier classes also, that a flower is having four whorls. Outermost whorl of green leaves called sepals, then variously colored leaves called petals, and male reproductive system called stamen, and the female reproductive system called pistil. Now if we see the parts of stamen, male reproductive system, it is made up of anther and filament. And here in between the two anther lobes, this filament is connected, and that part where filament is connected to the anther is called connective tissue. Then coming to pistil. Pistil is having three parts: stigma, style and ovary. Now in this chapter we are to take up events in sexual reproduction. As in all organisms, the events in sexual reproduction are common, and these events are pre-fertilization events, fertilization events and post-fertilization events. So first of all we will take up pre-fertilization events, and pre-fertilization events involve gamete formation and gamete transfer. So here first of all we will discuss what structures are present in a flower before fertilization and what are their functions. So pre-fertilization structures and functions in a flower. In a flower, before fertilization, male reproductive parts which are present: stamen, microsporangium and pollen grain. And the female reproductive part before fertilization are pistil, megasporangium and embryo sac. Now first of all, stamen, microsporangium and pollen grain in detail. Stamen is made up of two anther lobes. So we can say ki stamen is, or we can say anther is bilobed, because there are two anther lobes in stamen. So we can say anther is bilobed. And if we cut a transverse section of these anther lobes, we will see that each anther lobe, this is one lobe and this is another lobe. So you can see each anther lobe is having two compartments in it. This is first compartment, this is second compartment, this is of one lobe, and this is of second lobe, this compartment and this compartment. These compartments are called theca. So each lobe has two theca, or we can say each lobe is dithecous. So we can say anther is bilobed and each lobe is dithecous. And inside these compartment is present microsporangia or pollen sac, in which pollen grains will be developing. So anther is bilobed and each lobe is dithecous. See the spellings: D-I-T-H-E-C-O-U-S. So theca means compartment. So total microsporangia in anther are 4. So anther appears to be a tetragonal in shape. Now you see here TS of young anther, and then we will see the structure of TS of dehisced anther. Dehisced anther means split up anther, through which the pollen grains are being released. So, TS of young anther is made up of 4 wall layers from outside to inside. Let's see. Outermost, epidermis. Then second one, endothecium. Remember, it's not endodermis, it is endothecium. Then next is middle layers. Middle layers is 2 to 3 wall layers thick, and epidermis and endothecium, these are single layered walls. Then inner to middle layer is tapetum. So 4 wall layers from outer to inside: epidermis, endothecium, middle layers and tapetum. Tapetum is surrounding the microsporangium. Tapetum is surrounding the microsporangium, in which microspores will be formed, and these microspores will later on develop into pollen grains. So in anther, which event is occurring during sexual reproduction before fertilization? Formation of pollen grains. Then you can see these are 4 microsporangia which give anther a tetragonal shape. And each microsporangia is having sporogenous tissue. You can see these boxes. These boxes means these are all filled with cells. These are cells. So sporogenous tissue we can say, and each cell of sporogenous tissue is called microspore mother cell. Each cell of the sporogenous tissue behaves as microspore mother cell. And as I already told you that mother cells means diploidy. So these microspore mother cells are diploid in nature. Then you can see in a transverse section there is connective tissue. It is the tissue where the filament is attached to the anther lobes. So connective tissue is the position where filament is attached to the anther lobes. Now TS of dehisced anther. Dehisced anther means the anther has split up and pollen grains are released. But before that, I would like to tell the functions of these wall layers. Outer 3 wall layers: epidermis, endothecium and middle layers. First 3 wall layers are involved in dehiscence of anther. These are protective in function also. These 3 layers protect the pollen grains which will be formed inside, and also these 3 layers help in breakdown of the anther lobes to release pollen grains. And the tapetum which is surrounding the sporogenous tissue, here later on microspores will be formed, and from microspores pollen grains will be formed. So this tapetum is providing nourishment. Tapetum is providing nourishment to the developing pollen grains. So again, there are 4 wall layers. Outer 3 wall layers help in protection and dehiscence of anther, and innermost wall layer tapetum provides nourishment to the developing pollen grains. Now this is the structure of a TS dehisced anther. As I told you, 3 wall layers are involved in splitting of the anther to release pollen grains. So you can see here the wall layers have broken, and the tapetum has been used up by the developing pollen grains. So no tapetum is seen here, and pollen grains are released. Now in exams, they can ask you draw structure of TS young anther or draw structure of TS dehisced anther. So you have to see in the question what is being asked. Is it the young anther or the dehisced anther? So accordingly you are to draw the diagram. Now inside this microsporangia, as I told you, microspore mother cells are present. These microspore mother cells will be dividing to form microspores. These microspores will later on develop into pollen grain. Actually, it is the pollen grain formation, right? So this event is called microsporogenesis. Microsporogenesis. Where this microsporogenesis occurring? Microsporogenesis is occurring in each microsporangia of the anther. Now, what is microsporogenesis? It is the formation of microspores. It is the formation of microspores from microspore mother cell through meiosis. Microspore mother cells are diploid, and when they undergo meiosis, meiosis is a reductional division. Microspores which are formed are haploid. Chromosome number will be reduced to half. So where are these microspores formed? Microspores are formed here inside the microsporangia or pollen sac, and these microspores are formed from microspore mother cells, and this phenomenon is called microsporogenesis. Now you see here, microspore mother cell, a single microspore mother cell, when it will undergo meiosis, it will form 4 microspores, and these 4 microspores are arranged in a tetrad. One is behind these 3, so it is a microspore tetrad, and each microspore is having n number of chromosomes. Each microspore is haploid because it has been formed after meiosis. So, when all the microspore mother cells have undergone meiosis, so here what will be seen? Microspore tetrad. One microspore mother cell will be forming 4 microspores. Now, once these microspores are formed, these microspores, they will mature into a pollen grain. So this is a microspore tetrad. These microspores, they will separate from each other, and each microspore will undergo these events to mature into a pollen grain. So let's see what are the events here. Microspore increases in size and vacuolation occurs. Then this microspore will not undergo mitosis, it will not undergo meiosis. There is actually asymmetric spindle formation in the cell, which will result in formation of 2 unequal sized cells. Smaller cell is called generative cell, and the bigger cell is called vegetative cell. And this cell is surrounded by outer spiny layer called XII, and inner layer is intine. So this whole structure is now called pollen grain. So pollen grain is having 2 wall layers, XII, intine, and it is having 2 cells, one smaller generative cell and the other bigger called vegetative cell.